Right. Oh, thanks, folks, for coming along. So this talk is going to be slightly different from some of the other ones we've had, but hopefully you'll find it interesting. It's some research that I've sort of been involved in the last few years, which I found really interesting. And I've got a student working on some of this stuff at the moment, and I'll show you what he's doing. Yeah, the title comes from, there's a famous quote by James Dean in one of his movies, which actually came out of the book, the reference there, and it says, "Live fast, die young, and leave a good-looking corpse." So I've twisted that around slightly, and I'm going to talk about these things called ephemeral species, but more on that later. And I'll show you a photo of that. I'll describe what these are a bit later as well. This is work that's been done with my. Well, in parts of this, I'll describe bits and pieces done by my student Callum Whitten, who's close to finishing up, and by my colleagues Roger Cooper, Michael Foote, and Pete Sadler. So, right, just I'll give a brief overview of the sort of problem that I'm talking about. A big problem in paleontology and biology. And basically, one of the key questions, I guess, in paleontology, well, over many decades, is how fast do new species form and evolve? And it's a really fundamental question, and there's been a lot of work done on it. And one component of that, the problem is that if you try and measure the rates of evolution in living organisms and in fossil organisms, you come up with a very different answer about how quickly species are evolving and how quickly new species are forming. And it's quite hard to reconcile what you can see in the living things today and what you can see in the fossil record. And there's a real problem with the timescale we can observe, either in the fossil record or in the living, and actually the timescales at which things are actually happening. And I'll explain all this a bit more. Evolution is actually messy. We like to have these nice, simple models of what's going on, but it's actually quite messy. And I'm just going to talk about two examples. The example of Pelecaria, a lovely little endemic fossil snail. Well, it's still living today, and that's worked with my master's student, and an example from the graptoloids. And that's where I'll talk about these ephemeral species, but it's all linked together. And then I'll give a few conclusions. Okay, this is a classic view of evolution and speciation as paleontologists like to depict it. And that's just a lovely trilobite from the Smithsonian collection, remarkable thing. But anyway, so paleontologists have classified, a lot of paleontologists now would classify evolution and speciation like this. And on the horizontal is just some measure of evolutionary change. And on the vertical axis, the y-axis, is a measure of time. And so you can have just continuous gradual evolution. You can have continuous gradual evolution with what we call cladogenesis, splitting of the species pool. So you go from one species, at this point you've got two species, and they're both showing continuous gradual evolution. The alternative, and it was Eldridge and Gould who sort of championed this model in the late 1970s, was this idea of punctuated evolution. They said that actually species through time stay more or less the same for long spells of time. And then when the speciation happens, when the evolution of one species from another happens, it happens very abruptly. So you have what they called stasis, you have a punctuation, and then you have stasis again. And again, that can involve either just one lineage changing through time, or it can involve this cladogenesis, this splitting. So you go from one species and you get a split and you end up with two species evolving, but showing this stasis, and then another one. And so this is how classically how paleontologists, if you ask a majority of paleontologists to come up with a simple depiction of evolution, this is what they'd probably come up with, or something like that. And biologists get to see evolution in a very, very different way. And these days, most of that comes from studying the genes of organisms and comparing the genetic sequences and actually looking at the changes in the DNA from one organism to another. This is a lovely photo. This guy was studying bacteria, evolution in bacteria, and each one of these petri dishes contains, well, I don't believe they actually do, but it must have taken him ages to stack those up. In theory, each one of these contains one of his samples that he studied these evolutions. And here, and this is now somewhat out of date. This is figures from 2013, and the experiments have kept running since then. So these numbers would all be a lot larger, but 58,000 generations. It was running for 25 years, etc. You can read that, a very large number of bacterial cells. So this is studying evolution in a single-celled organism in this case, but in the modern. And you come up with a very, very different view of evolution than you do if you're looking at the fossil record. And you can characterize those differences in a number of ways. The way I'm going to highlight here, if you've got a simple graph of the evolutionary rate you measure, and you can measure that any way you like, you can look at changes in genes, or you can look at change in the size of an organism, or change in how many spines it's got. Whatever it is, you've got some measure of evolutionary rate, and you've got the time span of your observations. So that might stretch. You might observe changes in bacteria over a matter of days, or you might, at the far end of the scale, you might be looking at changes in groups of fossils over tens or hundreds of millions of years. And so if you look at these things, and if you measure evolutionary rate on a very short time scale, you tend to end up with a very high evolutionary rate. And as you get towards measuring it over longer and longer time scales, your evolutionary rate tends to go down, and so on. So you end up with something like that. You end up with this relationship that if you measure evolution on a short time frame, you get very high evolutionary rates. And if you measure evolution on a very long time scale, you get very low evolutionary rates. And you get a pattern like this. Now, I will just say, in part, this is actually a mathematical inevitability, but there's also a real story here. It's telling us something about how we are perceiving evolution, or about what it's actually doing in reality. This is showing exactly the same thing, but in a slightly more complicated way. This is based on a very famous graph that was produced by this guy Gingrich in 1983. But in fact, it was redrawn for a newsletter article recently, and they just prettied it up with some pictures. But basically, again, we've got time and we've got a measure of evolutionary rate, and the scale on these two things don't matter. But again, and what each of these little numbers on here says, how many studies were that measured a rate about there. So, here there's one study, and there's two studies that measured that rate, and so on, over these different time frames. And it's showing, these are the rates you sort of measure in a petri dish with bacteria, and these are the rates you might measure with a dinosaur or an ammonite or whatever. Just for those, this is the time, because it's spanning such huge amounts of time. It's on a log scale, so that represents 300 days. And at the other end of the scale, you've got 400 million years. A log scale of 0 means 1 million years. So that gives you some idea of the time spans. So again, there's lots of studies, the paleontological studies that come up with, they've measured over long time frames, and they come up with very slow evolutionary rates. And there's these studies of biological systems up here that measure over very short time frames, on a matter of days, and they come up with very rapid rates. And one of the things, there's many things in this diagram, but one of the things we see is there's a big zone in the middle here where there's essentially no studies. We've got no data. We've got a bunch of data up here, and we've got a bunch of data down here, and we've got nothing in the middle. And this is like this sort of valley of death, no observation. And it corresponds to a time span of several hundreds to tens of thousands of years. Over that time span, it's really hard to get data on evolutionary rate. And I'll go on in the next few slides and try and figure out why that's so difficult. But just before I show why that's so difficult, it turns out that actually is probably the critical timescale when probably an awful lot of speciation, an awful lot of the evolution that we'd like to be learning about, probably happens on the scale of many hundreds to a few tens of thousands of years. That's when we want to be knowing what's going on. And it's this time frame that it's very hard to observe. Here's a remarkable example based on living organisms. And these are cichlid fish. There's lots and lots of species. They're popular aquarium fish, some of the different sorts. the lakes in the African Rift Valley. So Africa is tearing apart along the Rift Valley through here. So the two halves of Africa, well, not the half, this part of Africa is moving away from that part of Africa. And as it pulls apart, the Rift Valley's been forming and these lakes have been forming. And we know exactly how old those lakes are. And so there's several lakes, but the one I'm going to focus on here is just Lake Victoria. And we know that that lake is less than 150,000 years old. We also know by looking at the genetics of all the cichlid fish that all these species originated with one species of cichlid fish that invaded that lake about 150,000 years ago. And there's now, that figure, it says 500, it's actually now out of date. We now know there's more than 700 species of cichlid fish living in Lake Victoria that have all descended from that one ancestor. And the same happens in Lake Tanganyika and Lake Malawi and so on. They've all done the same thing, these vast numbers of species, but that's happened over 150,000 years. And that sits fair and square. And we know that from the genetics, but looking at the genetics, we still can't actually observe how those species are forming. We can sort of make some educated guesses and we can do all sorts of, we can try to figure that out, but you can't actually see it happening because it's in that really awkward time span. Yeah, that was just to remind me about the time span. So why is this hard to see? Well, we obviously for biological experiments, we can't run experiments over 10,000 years. We can't do that. We can run them over 25, 30, 50 years if you're lucky, but that's about it. At the other end of the scale, the paleontological record, there's a simple limit in most of the paleontological record. There's a limit below which you cannot tease things apart. Even take just a shell bed like this. This is about 2 million years old. It's over in southern Wairarapa. These are all scallops. They're still alive today. Actually, it's the queen scallop today, but Cycloclemys delicatula, and there's barnacles and all sorts of things in here. But a fossil shell bed like this is an accumulation that may have accumulated over many thousands, tens of thousands, or possibly even hundreds of thousands of years. So if you collect a fossil from that, you're not going to be able to resolve that species, a fossil there changing into one there. You don't know the time span that these have been accumulated over, but it's likely to be very long spans of time. So you can't resolve things that are happening on the span of a few decades or hundreds of years because it's all mixed up in a shell bed that probably represents an accumulation of tens or hundreds of thousands of years. And this is a lovely illustration. I just saw this at Ototoka Beach a few weeks ago, actually, when I was taking the students up there. This is sitting up. This is completely unedited. This photo, I didn't manipulate this at all. This is on the beach. There's lots of fossil shell beds sitting around the beach. Those individuals are probably a few hours to months old, and that one is somewhere between 1 and 2 million years old. And the beach is littered with these things all mixed up. When this sand is buried and turned into sandstone, it'll contain a mixture of fossils that might vary in age by up to 2 million years. It'll be a mixture, so that limits the resolution of information you can extract from that. If you're working on microfossils, the problem's even worse. This is just an example of a Miocene formation up north, and the burrowing mixes up fossils of different ages. So again, that limits how finely you can study what evolution is doing because the stuff's all been mixed up. Here's another example. The fossil record is notoriously incomplete. And I mean, Charles Darwin worried about this enormously. But this is a complicated diagram. The details don't matter at all. But what it shows on the vertical axis is from the modern day going back 40,000 years. And the horizontal axis is actually distance. So there's a 10-kilometer scale. You get some sense. So that must be, what, a couple hundred kilometers wide. And the colored bits are the ages where there was rock deposited, and the white is where there was no rock deposited. So what we see is that the rock record here in this example is incredibly incomplete. What the geologists see is shown at the bottom. That's a geological cross-section, and they just see rock stacked on rock stacked on rock. And unless you have some very good ability to date those different layers of rocks, you're just going to think that's a more or less complete succession of rocks. But in fact, it's got huge gaps in it. So if you're trying to study in great detail the evolution of fossils coming up through here, you're actually going to have a little fossil sample here and then a great big time gap and some there and then a great big time gap. So you're not going to get that complete record. So again, it's incomplete. That limits the resolution you can extract from the fossil record in many places. And we don't usually, I have to say, it's not very often that you can actually tease apart the precise ages of the rock in this much detail. This is a remarkable and unusual and very lovely study. So even, I mean, what I've done lots of field work, you go out and you're doing detailed study. In this particular example, we were studying the change in the environment through one of these what they call the hyperthermals 50 million years ago, when there were these very abrupt and dramatic warmings on the planet. And people have been studying those for several years to try and figure out why did they happen, but also how did the planet naturally fix itself after one of these abrupt warmings? So what caused them and how quickly did they fix? But anyway, there's these lovely layers of limestone. This is at Branch Stream in Marlborough, a stunning spot. And we lay the tape measure out and we collect samples in every layer, every bed. And we do as detailed sampling as we can get. But every boundary between every bed here represents some time gap. Some time is missing. So it's not a continuous sequence. There's little gaps all the way through that. And we don't know what those gaps are. We don't know whether the gaps are 10% of the time is represented by gaps or whether 90% of the time is lost in these gaps. We don't know that. So even though we do incredibly careful, detailed sampling, you still don't know how you're not getting that really fine resolution that you probably need to study evolution at the scale that it happens. So bringing all that together, from the fossil record, you can study what's happening at everything from 100 million years down to timescales of maybe 100,000, maybe 10,000. It's getting pretty fuzzy if you've got a particularly good record. And if you're looking at the biological end of things, you can study things over years, a few decades if you're lucky. Maybe a century if you're really lucky. And then there's this gap, this observational gap in the middle that it's very hard to get the data for the reasons I've been explaining. So that's one of the big problems we have when we're trying to understand why evolutionary rates are so different between biology, when you observe them in biology and observe them in the fossil record. That's one of the reasons. And I've just, this one I just summarized as evolution is messy. This I showed you, this is the sort of paleontological model. This is what we'd like evolution to look like. Well, in fact, it's nothing like that. It's complicated. So for example, it's not either that or that. It'll be some combination. So as things go through time, sometimes they'll be evolving quickly. Sometimes they'll stay the same over long periods. Sometimes they'll branch and change quickly. Sometimes they'll branch and not change not so quickly. There's all sorts of things going on. And what this bit is showing is that actually a lot of the time there's probably a species probably consists of a series of populations that have been separated for whatever reason. They're separated out. They're not interbreeding. There's no genetic flow with the populations elsewhere. And if we're sitting at one time, if we're sitting today and we see one of these things, we might say, oh, that's another species. But actually, over time, and this is it's becoming increasingly obvious that this is very common, that what we might think of as a separate species gets resorbed back into the ancestral population through hybridization or other mechanisms. But so at any time, you've got this whole sort of mishmash network of what might be species, what might not be species, all mixed up. So the total picture is that evolution is messy, and that makes it difficult to try and understand what's going on. And I'll show some other examples of this messiness in a few minutes. In fact, I'll show them right now. Hold on, I just need a drink. So people have been doing a lot of trying to figure this out and trying to figure out, and for example, there's been a huge amount of debate of whether most species stay the same for a long period of time, show stasis as Eldridge and Gould proposed, and then change to a new species abruptly, or whether that sort of continuous slow evolution is very common. And people have come up with various fancy mathematical ways to try and figure this out. But one of the things that's come out of this, so in all these 4 plots, this is time. It doesn't matter what that's measured in. I mean, as it was originally said, it was generations, but it could be years. It doesn't matter. And if you make some very detailed observation, what you find is that things are changing the whole time. These little zigzag lines, they change a little bit one way, they change a little bit back the other way. And any species is just evolving rapidly the whole time. And you can fit mathematical models. And so even if there's continuous, what we might call gradual evolution going on, like with these 3 examples here, even then there's at a finer scale, there's the species are showing this rapid backwards and forwards evolution. And so people can fit models, and to a set of data, you might measure some aspect of your organism, as I say, the size or the length of its thigh bone or how many spines it's got or whatever. You might measure that through time and try and figure out, well, which of these models it's fitting to. And you can, as you do that, you're making assumptions about how rapid these fine scale fluctuations are versus the long-term trend. This one is called stasis because although it's fluctuating around, the mean is staying exactly the same over time. So it's actually the mean is not changing, although it's fluctuating around that, as we would expect natural things to fluctuate around. In this, they've called it anagenetic, or this gradual series. Again, the fluctuations are happening, but over time, it's changing. The mean morphology is changing. Random walk is the case where things actually are just randomly changing. They're not following a trend. They're not staying the same. They're just randomly fluctuating with no particular pattern, no particular rhyme or reason that we can discern. And actually, probably a lot of evolution happens like that. And this is an example of stasis, although again with the fluctuations, and then a punctuation event, and then stasis. But all these things can be described mathematically. And this is one of the tools people are bringing to bear to try and understand how evolution is happening in the fossil record. So for example, to try and figure out when these very rapid fluctuations are the thing that biologists observe today, whereas the long-term trends are what paleontologists see. And so, as I say, so it may be that what we're seeing up here, going back to this diagram, are these rapid fluctuations, a thing reacting very quickly to a stormy year this year, an El Nino for the next 3 years, and then a drought the last 5 years. So they're reacting backwards and forwards. That's what we're observing here. Whereas what the paleontologists observe is the long-term trend over long spans of time. So let's just look at a couple of examples. And this is my master's student, Callum Whitten, who's in the last month or so of finishing up his master's, and he's done an absolutely stunning bit of work and working at these lovely little snails. A thing called Pelecaria vermis is still alive today. It's a deposit feeder. You can pick it up on some sandy beaches, but it's endemic to New Zealand, which makes them a lovely study group. They lots rich fossil record over the last, well, I suppose how far back do they go? 15 or so million years, I think. And they show huge variety of form. This is just 3 randomly chosen specimens, but they show great variety in size, in the form of these ribs, and different people over the last 30 or 40 years. Some people have split them into many species. Other people have said, no, it's all just one variable species, that and others people have said that it is one variable species, but it shows systematic differences. It looks different depending on whether it's warm climate or cold climate. So there's various things to test, and Callum has been studying this. One of the other lovely things about these snails is they're what we call determinate growth. So they reach, a lot of snails just keep on growing, and they might keep changing as they grow, but they never stop growing. They never have a fixed sort of adult growth stage. Whereas these things are great. When they've reached adulthood, they stop growing. They put on a great big thickened lip here, and then you know you've got an adult. So it means if you're trying to compare them through evolutionary time, you can just pick the ones that've got this thickened lip, and you know you're comparing them. You're comparing adults with adults. You're not, say, comparing adults with children, which might be different for a whole another reason. So that's one of the really nice things to do. Really nice aspects of these snails. So Callum has collected data from two main areas, from up the Mangahao River in southern Hawke's Bay and from the Tamuna area in southern Wairarapa, and has logged these hundreds and hundreds of meters of section, collecting these things in populations all the way through, so he could see how the different layers, the populations in the different strata compare to each other. I'll just note that one of the other appeals of this group is that, so Jerry Neef in the 1970s did his PhD in this area, and he described a number of species, but he also thought that the species stayed the same for long periods of time and then showed punctuated evolution. And in fact, Eldridge and Gould, in their original description of punctuated evolution, criticized his approach, and for reasons we don't need to go into. But the fact that they picked on this, and it's so also makes it a lovely study example. You know, did Neef have it right, or were Eldridge and Gould correct to criticize what he'd said? So to do this, Callum's doing some really fancy stuff. Actually, so actually another former student of mine recently published a paper on how do you quantify the shape of a snail. It's actually surprisingly hard to do. Snails are really hard to come up with a set of measurements that are useful. There's a famous study by a guy called David Rope who came up with just 3 simple descriptors that describe how a snail coils, how it grows as it coils. When it coils, so if you cut a snail down the middle, it would look something like this, and you've got a series of apertures opening up and growing, and you can describe it by how quickly those apertures, how far apart they are in the vertical sense, how far out each time they grow. Each new aperture grows from the coiling axis. And sorry, brain fade. What's the third one? Oh, I've got them listed in a second. I'll get onto that in a second. So I'm just having brain fade. But this obviously is a very hard thing to measure in a snail often, and particular in some of these things like Pelecaria, that often the adults get completely covered with a big thick callus. And this is an image a student of mine, Katie Collins, took on X-ray image showing the interior cavity within the snail completely hidden inside the callus. And obviously measuring these sorts of things on a shell like that, if you haven't got X-ray vision, is extremely hard. So what Callum's done is he's cut a bunch of these things up, having collected them painstakingly. He's sawn hundreds, or about 282, I think, in half, so he can look at how their coiling is changing. And he's using some fancy maths to describe this. He's looking at the rate at which, this was the one I couldn't remember, the rate at which the apertures are growing between each whirl, how quickly they grow, the rate of the rate they move downwards, and the rate they move outwards. He's also measuring the shape of each aperture and how that changes the rate of change of the aperture. In addition, he's looking at so these are all based on the interior cavity of the snail, but he's also looking at the form of these ribs, the different ribs, and I showed you on the previous slide how varied those can be. So looking at the form of these ribs using a different sort of maths. And finally, he's looking at size. So he's got all these different things. He's looking at how these different things evolve from one population in one layer of strata to the next population, and so on through time. And what he's going to be doing, and I'm not going to present any results, I'm not going to steal his thunder, that will come at the Geosciences conference, hopefully at the end of the year. But here's an example of another study that was done on bryozoans, fossil bryozoans. And this is a famous study done in the 1980s. But this person made a series of measurements, and it doesn't matter what he was measuring, I can't even remember, to be honest, but he measured a whole series of things going up through time. And he said, well, these clearly show punctuated evolution. They stay the same, and then there's a sudden jump, and you can see, they stay more or less the same, and then there's a sudden jump, but these remarkable periods of stasis. So Callum is going to be doing a very similar thing, using those mathematical models, using these models to figure out whether they're showing punctuated, whether they're showing stasis, or whether they're showing the continuous change or random walk. And in fact, without taking too much from him, I would say he's finding pretty much all of these in different characters. The things, one character is evolving one way, a different character is evolving a different way. So, size does one thing, whereas the shape does another. And what he still has to test is whether some of those are being driven by environmental change. And so for environmental change, he's looking at the grain size of the sediment, because it's a really useful general measure of what the environment is doing. So that really is just an example of the sort of work that can be done, and trying to figure out how this evolution is happening. I'm now going to get to the ephemeral species, the title, and talk about a bit of work that has been done. I can show you some results. And this is using graptoloids. So for those who don't know, graptoloids were the major zooplankton group. So animals floating in the water column through much of the Paleozoic, for a large chunk of the Paleozoic. They comprised, they were a colonial thing. So that slab of rock is from Northwest Nelson, from Kahurangi, and that's about 15 centimeters wide, to give you a sense of scale. And each one of these is a graptolite. There's several different species. That's one species, that's a different one, this is a different one. There's several species on that slab. And so each one of these was a colony. Here's a zoom up of the colony. It comprised a series of cups. Each cup had a little animal living in it. They all sat linked together, and they were suspended from floats like this. And the floats are hardly ever preserved, but these parts are preserved. And through the Silurian and the Ordovician and Silurian, these things were essentially the most abundant animal floating in the ocean. They completely dominated the zooplankton in the ocean. Ecologically really important, and they evolved really quickly. And it turns out they're really good for biostratigraphy, which is why they've been studied so intensively. And Roger Cooper, in fact, originally did his PhD on the graptolites from Kahurangi National Park. Because they've been used all around the world for over 100 years to do biostratigraphy, our knowledge of their fossil record is incredible. And this, I can't claim any credit from this. This is over about 12 years or more. Roger Cooper and Pete Sadler, who's a scientist in California, compiled this incredibly detailed record of the global fossil record of the graptolites. And this diagram, it's called a spindle diagram. It just shows the number of species through time changing as it fluctuates, but very, very high resolution record. And it's colored by the different families, the different types of graptoloids. This is the mass extinction at the end of the Ordovician, when the numbers plummeted. But not only did the numbers of species plummet, but also then the composition, the ones that took off after that, were completely different groups shown by the different colors. So it's amazingly highly resolved fossil record of graptoloids. It's a global record. These dots show all the places where they've compiled data from. This is just for the 460 million years, the Caradot stage. But it shows you that very obviously the geography doesn't look anything like it does today, but shows you that this is a very an incredibly complete data set. And from their analyses, and I say a huge compilation, we've got very detailed record of the precise age ranges of 2,045 graptoloid species, going from about about 490 million years ago to about 410 million years ago. And each one of these bars represents the duration of one species. And as I say, there's 2,045 bars in that. That just shows some details of the database. I'm not going to bother going into that. But one of the cool things, another colleague of ours, Michael Foote, did some very clever mathematical modeling on these. And he reckons you can demonstrate that about 75% of the once living species have been sampled in this database. So of all the species of graptoloid that were living through this interval of time, he says about 75% of them have been captured in this database, which for a fossil database is absolutely remarkable. For fossil birds or fossil mammals or whatever it is, that number would be, you'd be down at Well, I don't know. I'm completely guessing, but if a few percent of the species that lived have we actually got fossils of, whereas these graptoloids, because they were floating in the ocean and they lived everywhere, they have an amazing fossil record and they've been very, very intensively studied. So we've managed to get this amazing fossil record. Now, this is a bit of a nasty graph. I'll walk you through this. It's quite instructive though. So what this shows, both this one is just an enlargement of this part. So let's just focus on the left at the moment. This just shows how many million years species live for. So 0, 5, 10 million years. And it shows the proportions of the fossils that live for each period. So what we can see is for the Silurian, the red line, you know, about, what's that? Must be about 2% lived for over 5 million years. Whereas for the Ordovician, actually, 2% lived for over 10 million years. But you can see, so about 10% lived for about 2.5 million years, if we read that off and so on. If we enlarge this bit up, the key thing, what I want you to draw your attention to, and this is just built on those 2045 ranges I just showed you. That's comes straight from that. What we see, if we just enlarge up to this bit, what we see that actually this is 50% of the species lived for less than 500,000 years. That's 500,000 years, about 50% of the species lived that long. The 50% lived slightly longer in the case of the Ordovician. The Ordovician species tended to live longer than the Silurian species, obviously. But just hold that thought. So 50% of the species lived for less than 500,000 years. And in fact, you can come up here, and that's 70%. 70% of species lived for less than 100,000 years. They're really, really short-lived. These things, they weren't living for millions or tens of millions of years, which is what we tend to think about fossil species living for. These things were living for really short spans of time. And as I say, if you just hold the figure, 50% lived for less than 500,000 years, you get the idea. And we can only figure this out because we've got such a remarkable fossil record that has been so intensively studied. Now, again, this is a complicated figure. I'll walk you through this. So just ignore the graph for a second. What we see is that a high proportion of these species live for very short times. What we also see in this data set is that they were going extinct and new ones were evolving in lots and lots of little pulses, you know, blam, 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 blam. And when some went extinct, new ones evolved, and at about the same time. So there's a very strong correlation between extinction and origination of new species on a very short time frame. It was happening in the space of a few tens of thousands of years. A bunch went extinct and new ones evolved. And then a few tens of thousands of years later, a bunch went extinct and new ones evolved. And this was happening just like clockwork. And that's so what wasn't happening is that it wasn't that those two things, you know, some went extinct at one point and then a bit time later another one's evolved and then some new ones evolved and then some went extinct. These things were happening together and they were tightly coupled. And there's this very strong correlation between evolution and extinction. And actually I hesitate to this is just showing that correlation, which surprised us and was unexpected. So the correlation, for those who are interested, you know, is almost 0.5. This is what we expect that correlation to be, somewhere in here. We expect, for mathematical reasons, there to be a correlation between them of, you know, a 0.15 or something like that. We do expect that, but what we see is what we observe is so much more than that, and it's very highly significant. And that's all we need to take from that. So the point is that the evolution and extinction are happening in myriad little pulses very rapidly. So if we sort of bring all this together, and some other stuff which I haven't talked about, most species were short-lived, less than 500,000, and many of them much less than 500,000 years. Evolution and extinction of species were tightly correlated at less than a 50,000 year timescale. So they're happening together rapidly. We also know from other studies that I'm not talking about that the evolution extinction rates were being driven in part by Milankovitch cycles. So that's the name we give to these wobbles of Earth's orbit. There's very regular wobbles as Earth's going around the sun. It wobbles, and those wobbles cause regular variations in the amount of, well, where the sun hits the planet, and they have this effect on climate. And they're very regular, and we know that the evolution and extinction were being controlled by those to a fair extent. We also know that newly evolved species were more vulnerable to extinction than older species. So if you had two species alive together, one that had been around for 10 million years and one that had been around for 500 years, what we know is classic theory would say, well, those have an equal chance of going extinct. It doesn't matter how long you've been around. You have, at any time, you have as much chance of going extinct as the thing that's only been around for a short period of time. Well, in fact, we see that's not the case. We see that the species that are newly evolved much more vulnerable to extinction than the species that have been around for a long time. And the last thing, we also see that there's this thing called diversity, negative diversity dependence. And by that, I mean, and that happens over long time frames, over 1 million years. So what that means is if there's lots and lots of species living today, then 1 million years down the track, extinction rate will go up, or the rate of evolution will go down to bring that diversity down. But that happens over 1 million years. Conversely, if there's very few species today, for whatever reason, then 1 million years down the track, extinction rate will go down or evolution rate will go up to bring that number of species back up again. So there's this thing that the amount of species alive control the rate of evolution extinction some way down the track to try and keep the diversity around some sort of even level. And I'm oversimplifying here, but you sort of get the idea. Sorry, it's all quite complicated, but it tells a neat story. So this is the final model we come up with, and this is where we get onto the ephemeral species. So in this model, let's look at the dark gray box. You have species forming through speciation or evolution. You know, all these different graptolites forming here. And what we find is, and then a bunch are going extinct. And this is happening very rapidly. Short-lived species are coming and going, just blam, blam, blam. They're evolving and going extinct very rapidly. And to relate this back to what I said earlier in the lecture, think of the cichlid fish in Lake Victoria. If the Rift Valley in East Africa, if the tectonics change slightly or the lake dries out for whatever reason, all those fish will go extinct. Or a bunch of them, although we call them separate species now, some of them are going to hybridize with other species and disappear. They will go extinct for that reason. So a lot of those 700 species in Lake Victoria are probably what we would identify as ephemeral species. They're here today, and we can see them, and we can look at them, and we think they're different. But actually, on a geological timescale, they probably won't last. And that's what we're seeing in a lot of the graptolites. An awful lot of the actions happening on, a few tens of thousands of years, and species coming and going. What we also see, though, if the species manage to survive through 50 or 100,000 years, they escape that. And I should say these ephemeral species typically are found in one water mass or in one ocean or in one sea. They aren't found globally. If they manage to escape that, survive that period, and they manage to start dispersing to other oceans, to other water masses, then once they've made that, then they can stay around for 5 or 10 million years. They're much more resistant to extinction. And these are the ones that the long-lived species, and these are typically the things that we see in the fossil record for all species, whether it's mollusks or certainly for dinosaurs or birds or whatever it is. The things that we see in the fossil record are the ones that are not the ephemeral species. They're the ones that have escaped that and have managed to spread out to a reasonable, so they have a reasonable geographic range. They're found in many places, and they tend to survive for a long time. And it's these ephemeral species start to from the paleontological side, start to bridge that gap into that sort of observational gap between biological and paleontological evolutionary rates. So we're just going to finish off here. I guess basically what I'm saying is paleontological studies, yes, there's all sorts of problems that make it really hard from paleontology to get up into this time frame. But with the right fossils and the right databases and the right techniques, we can start to get up into here. And likewise, with biology, it's getting cleverer and cleverer ways of interrogating, say, the genetic code of organisms to come down into this. So we're slowly managing to bridge this gap and reconcile the paleontological and ecological views of evolutionary rate, if you like. And on that note, I've spoken quite enough. I'm happy to answer any questions.